I want to go ahead and just put in this introductory slide, put us where we are as we're going to start talking about Alzheimer's. Okay, and so Alzheimer's disease, like Korsakoff syndrome, so I didn't say this with Korsakoff syndrome, but I'm going to say it now, they have better procedural memory than declarative memory. If we go back and look at the brain structures that are involved, the basal ganglia appear to be fine in these people, and they're able to learn the weather task. Whereas we see Huntington's and Parkinson's disease patients, while we don't think of those as being memory disorders, that their procedural memory is what's impaired. So in these disorders, they have better procedural memory, worse declarative explicit memory. For Alzheimer's disease, it really, early on, it starts out with a great deal of variability in their memory and their alertness. If you remember going back to the neurotransmitters, when I first introduced them and talked about what the dysregulation of neurotransmitters can mean to us, it was acetylcholine and having low acetylcholine that was related with Alzheimer's. Well, low acetylcholine means I'm necessarily going to be less alert and have less attention, and it is very much influencing the hippocampus. We see smaller hippocampi in people with Alzheimer's. It's also related to memory problems, and again, correlational, but that's what we see. As we start out really not laying down new memories very well, we're going to see anterior grade amnesia more so than the retrograde amnesia of when we start to see that somebody has symptoms of Alzheimer's. We're going to see that they're having a real hard time laying down new memories. And then it's going to have that kind of graded appearance that we saw in that slide with the graded appearance where the Australian talked about his memory loss. But that graded kind of retrograde amnesia is going to move backwards in time. So a lot of people get Alzheimer's later in life, and we don't completely see that. I do have the early onset Alzheimer's in my family, and so I've seen several members of my extended family have gone from, you can tell they don't really have the memory and alertness. They have some confusion sometimes about where they are. They have memory loss, but as they have this for longer and longer, they start to lose more and more of those older memories. And in fact, my grandmother and my aunt lived to the point of not remembering how to eat, how to swallow, asking what am I doing, what's in my mouth, how to go to the bathroom by themselves. They can lose a great deal of everything, but it really starts out with that attention, alertness, and not laying down new memories, confusion, and it's variable. It's really variable of how often they're also sometimes lucid. So we see this memory loss, we see depression, restlessness. They sometimes have hallucinations and delusions. Of course, they're having sleeplessness, which can be very confounded with a lot of those things, and a loss of appetite. Your author talks about several theories, hypotheses of what's going on with Alzheimer's disease. Why do we see it? When are we more likely to see it? And what's happening in the brain? I'm going to start with this. Why do we see it? Why do we think it's occurring? And who's more likely to get it? So chromosome 21 is implicated in Down syndrome, which is one of the reasons they started looking to chromosome 21 and those particular genetics as potentially being linked to early onset Alzheimer's, as people who have Down syndrome, if they live to be middle-aged or have in middle adulthood, then they are very likely to have early onset Alzheimer's. So Alzheimer's affects 50% of the people over 85 years old and 5% of the people 65 to 74. And as we have this aging population, we're going to see more and more Alzheimer's. But about half the patients with late Alzheimer's, late onset, have no known relative with the disease. And we're not sure completely about the genetics of what's going on there, but it does not appear to be very genetically influenced. It looks like there are lots of epigenetic effects. I mean, it's genetic, but there's a lot of influences of environment And then I know I've seen one study looking at the APOA gene in people in Africa and people in African Americans in Chicago, and they all had the APOA gene, and their diets, how much sugar and saturated fats they were eating was very important for how likely they were to have Alzheimer's. But early onset Alzheimer's does seem to be influenced by genes, and it's much more rare. And what early onset Alzheimer's refers to is people who we see showing symptoms before the age of 60. One hypothesis of what's happening in the brain is that we tend to see higher amounts of amyloid beta, although that's not a really perfect correlation, where some people with early onset Alzheimer's have not as much amyloid beta, and some people with high amyloid beta don't have Alzheimer's. But what appears to happen is the amyloid beta cluster into plaques, and they cause damage. It causes damage to dendrites to axons, and so we're not getting synaptic input, and we're losing synaptic plasticity. And this causes regions of the brain to basically waste away. So areas of the cerebral cortex, the hippocampus, and other areas. And you can see over to the right the degeneration. So in A, that's a quote normal person's neuron, cortical neuron, and B, we see neurons of someone with Alzheimer's, where we are losing a great deal of the dendrites, the axon, and you can see the very end one has lost pretty much all aspects of any ability to communicate with other neurons. And what you see to the left, the A and B. So A is an example of a brain of someone who had Alzheimer's, and B is a quote normal brain. And so you can see that the folds are much deeper, and the sulci are much deeper because the gyri have lost just neurons. Another hypothesis of what's going on with in the brain in people with Alzheimer's disease is that it's the tau proteins that are not binding to their usual targets, and so spreading into cell bodies and to dendrites. And this is normally to carry information down the axon, and these kind of little almost trains getting the microtubules down to the axon. And so this causes these tangles, and tau protein, it does look like it's high levels of amyloid beta that are causing phosphate groups to attach to the tau proteins, which is causing these tangles, and so this neuronal damage. There is no known drug that is effective for treating Alzheimer's disease. One treatment they have tried is enhancing acetylcholine activity to increase arousal, as that can be helpful. Something that an avenue that appears to be promising from animal research has been to look at curcumin or turmeric, which is an Indian spice that has been shown to inhibit amyloid beta deposits and phosphate attachment to tau proteins. Well, I will say, so this, again, human research is just beginning, but I have a lot of concern for Alzheimer's in my life, as I have seen several of my extended family members die from Alzheimer's disease, and really have because it was early onset, have that slow, long downward spiral in their memory, attention, with depression, confusion, and all of that. And I have had, so one of my friends she actually ended up dying of lung cancer from smoking cigarettes her whole life, but she had, because of a car wreck, some body damage where she couldn't move for a little while, and a little bit of brain damage from the car wreck. And then she had the chemotherapy. And I know this is seeming off track, but there's a point to all of this. And so because of her, if you remember that we have nicotinic receptors that are acetylcholine receptors, and she was smoking cigarettes, that she was stimulating this acetylcholine her whole life with cigarettes. was unable to smoke anymore because of the lung cancer. And another thing that gets acetylcholine released is movement. And so she was also not really able to move anymore. So her doctor ended up prescribing choline, which is the amino acid precursor to acetylcholine. And it does exist in our foods. I've had one pretty really intelligent person in the field of neuroscience say we have lots of choline in our body, like we do tyrosine, and that we don't really need somebody to prescribe us choline, and we really don't need to concern ourselves with getting too much choline. But I have thought about this a lot, and so one of the things that has choline in it is farm fresh eggs. Farm fresh eggs, even more so than the mass-produced eggs, have choline, and we can use that as a precursor to acetylcholine. And a wonderful thing in that is to just throw a little bit of turmeric and pepper, which not only is the turmeric inhibiting these amyloid beta deposits and phosphate attachment to tau proteins, so potentially helping us potentially with Alzheimer's. Again, research isn't quite there yet, but I always have hopes. Also, turmeric and black pepper have a nice antioxidant effect, and so I also have lots of cancer in my family. So I do eggs with turmeric and black pepper quite a bit as one of my just hoping to stay healthy, and then yet still trying to watch my cholesterol and not eating too many eggs, just trying to stay healthy in the ways that I know are potential dangers for me, given my genetics and everything. Other areas of the brain that are involved with our learning and memory phenomena, again, I think it's really important to keep in mind mostly the hippocampus and the striatum and what their roles are. But we have also talked really pretty clearly about the amygdala in a different context because we talked about it in the emotion chapter, but its importance for classical conditioning is clear. People with parietal lobe damage, they don't appear to be able to elaborate on memory spontaneously. So usually when we're telling a story, what's happening is we are associating one piece of information with another, and we have this really elaborate kind of memory. And what's happening with people with parietal damage is they'll tell you something that happened, and in order to get those details, you would have to ask, well, where were you? Well, when was that? And they can answer those questions, but that kind of elaborating on memory is just not happening spontaneously. People with damage to their anterior and inferior areas of the temporal lobe have a semantic dementia. So this is really where it appears to be that our memory is for fruits, vegetables, animals, the things we've had experience with our lives that we know facts about, that information appears to be stored. People who have a semantic dementia might go into the grocery store and be very confused about where the vegetables are or what the word vegetables mean. So their language, that has a lot to do with language. Your author talks quite a bit about not knowing which animals, like what are those stripes on the horse doing, when someone's looking at a zebra, as we just lose that semantic information rather than the episodic information. And we also see areas of the prefrontal cortex are involved with decision making, our working memory, our memory of rewards and punishments, and then how that influences our more conscious thinking about our decision making. That memory is involved there. So lessons from studying many different kinds of people with amnesia. Much of the brain contributes to memory. There can be deficiencies of very different aspects of memory with other aspects of memory relatively intact, where we see these double dissociations. Even though, of course, they communicate with each other and contribute to each other, one can behave independently. One area can behave independently of another area and vice versa. So there are these very independent kinds of memory, and various kinds of memory are going to depend on different areas of the brain. And that's my overall trying to say as much as I can and keep it as general as possible, even though, again, we've talked about some of the really important areas and their roles. So I'm really hoping to finish this today. If not, I'll have a teeny tiny sliver for Monday. And we're going to come back to these questions because I realized that when I was talking about changes occurring in the pieces, I really started out with the regions of the brain, which are the larger, that are as much changes in the wiring in some ways because it's the neurons and what they're responding to, the particular stimuli they're responding to, but also the neurons in a particular area, and so how they're communicating with each other. So I haven't really distinguished those questions yet, and we're going to come back to distinguishing those questions. And so we're going to first start by looking at what's happening in a single cell, but we're also going to see the changes in the synapse, and so the changes in the wiring as well. Okay, so the nervous system has to be adaptive. It needs to be able to change if we are to learn new information and if we are to be adaptive. And so when we look at the storing of information in the nervous system, it's often discussed as Hebbian learning. Donald Hebb in 1949 was quoted as saying, "If axon of neuron A repeatedly or persistently excites neuron B, growth processes, metabolic changes, occur in one or both cells." And so a synapse that increases its effectiveness because the presynaptic neuron and the postsynaptic neuron are active simultaneously, that is going to be what allows us to be adaptive, and that's going to allow the nervous system to be adaptive. And so that kind of synapse, that when we have the neurons that fire together, wire together, so to speak, that's discussed as a Hebbian synapse because that is increasing the effectiveness of that connection that has been useful. This all makes sense as these kinds of synapses, as this kind of learning, as being involved in associative learning. So some of the early research conducted examining how we store information in the nervous system was conducted on the aplysia. This is a sea slug, and the sea slug has a very simple nervous system, very straightforward to examine. And we know that a nervous system is a nervous system is a nervous system. And once we know how a nervous system responds, we can extrapolate that to other animals and to other nervous systems. And I'm going to talk about habituation and sensitization as these kinds of non-associative learning. So in habituation, what happens is we decrease our response to a stimulus when it is repeatedly presented and accompanied by no change in other stimuli. We usually, in my classes, come up with some examples, and one example is the ding, ding, ding on your car that tells you to put your seatbelt on. And most of us just put our seatbelt on to stop it because it's a little bit annoying. But if you've ever just let it go, so I used to sometimes have my dog riding in the car and the ding would happen. And after a little while, you habituate. Okay, as we listen to something, we talk sometimes about the habituation of listening to the train whistle. If you live near a train, you eventually habituate and you don't notice as much that train going by as when you first move into a new apartment or whatever that's close to the train. So those are examples of habituation. What they did with the aplysia is they would have a small burst of air that went across that siphon. So this little area where they are sensing out there, and that little burst of air would at first cause this gill with the gill to withdraw. So this gill withdrawal reflex. And what they found is if nothing happened, so they would just keep puffing air past this siphon, that pretty soon that gill reflex would habituate and we would see it less and less. And basically, they also showed that this was not happening, that habituation was not happening at the level of the sensing or the feeling of that burst of air going by, and it wasn't happening because of fatigue of the motor neuron. What's happening is there is a change in the number of action potentials being sent down the axon of the sensory neuron and a decrease in the neurotransmitter that is released at the synapse between the sensory neuron and the motor neuron, and that's causing a habituation. Another example of non-associative learning is sensitization. And it's always, so this is when we have an increase in response to a mild stimulus as a result to previous exposure to more intense stimuli. So my system, my autonomic nervous system is already aroused, and so a mild stimulus can then cause later for me to have a more intense response to that mild stimulus. And I'm using the word stimulus and stimuli, which is not always helpful for people, and so I usually have students come up with examples, and unfortunately they usually come up with examples that are more on the order of classical conditioning, where the mild stimulus is predicting something like a shock. That is an associative learning. If I see the light and then I get a shock, I'm learning that association, and that is not sensitization. Sensitization is I was in a state of arousal when I heard the mild stimulus. And so sometimes use the example of PTSD. So if I was in a traumatic situation and I heard some kind of boom, boom, pop, pop, pop later on, when I hear that boom, 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 pop, pop, pop sound, whether it is fireworks or popcorn or whatever, I might have a really intense response to that because of the autonomic nervous system arousal when I experienced similar stimuli before. We might notice that people with autism spectrum disorder sometimes sensitize to things that most of us habituate to, and that's because a lot of the times their autonomic nervous system is aroused when they are experiencing these really relatively mild stimuli. What's happening there is, so in this case, what they did to the marine mollusk, to the aplysia, is that they shocked the aplysia first, and then they gave that burst of air to the siphon. And so what they would learn to do then is that when the burst of air to the siphon came, they would do the even a more and an increased vigor of the gill withdrawal response. And what's happening there is now, if you remember, if we go back to that picture before, their sensory neurons are communicating directly to their motor neurons. Nice, simple nervous system, but there will be a facilitator neuron that can also be communicating there with the presynaptic terminals of the sensory neuron. And so that facilitator neuron is going to release serotonin to those sensory neurons. That's going to block potassium channels. And so what's going to happen is that neurotransmitter from the presynaptic neuron is going to be released for longer. So we're going to see continued release of neurotransmitter from the presynaptic neuron, and then sensory neurons will synthesize new proteins that produce long-term sensitization. This is another slightly awkward place to stop and start, but there is just a tiny slice left of the structural changes in the nervous system that we see as a result of learning, and I will have to save that for Monday. I think I have run out of time. Everybody, I hope you are all doing well and that you have a great weekend.